Hello, I am Dr. Freitas, and I'm going to share with you my research, potential role of T cells markers in the autoimmune response. And this is a research that has been done in collaboration with Brigham Young University, Department of Microbiology and Molecular Biology. Now let's start, and I will say would be a review for most of you of what is periodontitis. But in order to ask what is periodontitis, I would say, why do we even consider still periodontitis a public health problem? There is a huge chunk of the population, not only in the United States, but also around the world, that is suffering of this disease. This is also a toll in the government with issues because this is a type of disease that would affect other systemic diseases in an individual. So targeting and understanding different ways that could help in improving the health of patients that are already developing periodontitis, it is relevant. To understand how immunology could help in the process to improve periodontitis, it is important to understand all the processes that are involved during periodontal disease. And in fact, a lot of research has been funded just because this is becoming an important project, a priority to investigate and improve patients' health. If you see, when we talk about periodontitis, we are looking into the building of the biofilm that would trigger immune responses. Those primary immune responses are the ones that we know as innate immune responses. Then would trigger different molecules like cytokines and chemokines that would attract other cells, and cells that will then, like ABC cells, that will help in the lymph nodes to activate lymphocytes. These lymphocytes then will contribute to control other cells, contribute to the inflammation process, and making it chronic, producing other molecules such as prostaglandins, metalloproteinases, and round ligands that will contribute to the recession of this tissue and also the recession of the bone, which are associated with the advanced damage of periodontal disease. Now, what is T cells involved in all this process, and what is important to trigger their function? T cells act like a balance during the oral health and disease. We have two main populations, Th17 and Treg cells. They're supposed to be in balance to keep that normal immune response in our mouth, in our oral cavity. If there is a type of disbalance in which the population of Th17 cells increases, then we will have the activation of more cells like neutrophils that contribute to inflammatory processes right here, and then the production of other molecules of ligand that will contribute to the bone resorption and inflammation that we'll see in periodontitis. Because of the importance of these populations, we were wondering how can we do to modulate T cell responses in periodontal disease. So we came with a hypothesis. First, let's see how the different proteins that appear on T cells contributing to T cell function. We have these different molecules, CD28 that contribute to activation. We have PD1 and CTL4 that will control the activation process, sometimes in a negative way. Those are the ones that are more known. Now, there is another protein called the CD5, which is shown to modulate T cell activation, not only on T cells, but also in B cells. So we wanted to explore how this protein can actually modulate perhaps the activation or the function of T cells. So our hypothesis is that surface markers in T cells, like CD5, would influence the activation response when exposed to microbial challenges such as LPS or lipopolysaccharide from P. gingivalis, which is a pathogen associated to periodontal disease. In order to recreate that, we try to recreate the microenvironment that we have in the oral cavity. For that, we got We are using cell lines from oral mucosa and gingival epithelial cells from mice, and we are using a specific mice cell line, which is the C57 black 6 mice. The stem mice are used to isolate T cells to make sure that there is no other variables in this experiment. The activators were using LPS for not only P. gingivalis, but also E. coli as a control. And we also something called dynabits, that are bits that will contribute to the activation. They trigger the activation signals important on T cell activation. So we're using, those are called dynabits as an activator. And also we're using T cells. More specific, we're looking in the population of T helper cells. And we are isolating them from the spleen of these mice, the wild type, and CD5 knockout. Knockout means it does not have the CD5 protein of the T cells. And we look at different time points to see how we can evaluate the function of the T cells and see if the CD5, in fact, is influencing the activation and other functions of T cells. So what I'm going to show you in the following slides are associated with the preliminary data that we have. So this data is going to be showing an activation, differentiation of T cells, cytokine production, and then the pro-inflammatory transcription factors. Now, the activation is important. Once the T cell is activated, you remember it has two signals. It has to have the MHC activated with the TCR complex. This is one signal. The other one would be the CD28 with another one from APC cell, which is the CD886. And then finally, we have cytokines that contribute to the activation of the naive T cell to an activated T cells. Also, once it is activated, to see how fast it is activated, there are two molecules that will give us that, and that would be the CD69 and the CD25. CD69 is activated, right? The T cells being activated, it's a protein that will show up in the surface. So we measure if there is a differentiation of CD5 molecule in the activation process of these T cells. And what we observe is that actually, yes, CD5 will regulate T cell activation responses at 24 hours and even at 48 hours. Even though in some of them there are no significant responses, overall you can see that even gingival tissue and the mucosal tissue, the wild type CD5 T cells have a higher activation compared to the CD5 knockout. We then went and look into the differentiation, looking in these different populations that I mentioned before, the Tregs and Th17, that once they are in balance, then we should have periodontal health, no any problems with bone resorption or tissue or recession of the gums. But then as we see that we have the Th17 of inflammatory T cells increase in number, then these are going to contribute to the inflammation process and the destruction of the bone. So we were looking into that and wondering if the CD5 could control that differentiation, looking that after 5 days of exposure to the cytokines, the activators, the lipopolysaccharide. So we measured again the gingival and mucosal from the Th17 being the wild type, the one that will produce more Th17 compared to the CD5 knockout. Same thing with the wild type producing more Treg cells compared to the CD5 knockout, mentioning, yes, CD5 can modulate and control these differentiations of T cells. Then we were looking into cytokine production. If you remember, we talked about pro-inflammatory, meaning they will support those inflammatory processes that happen in periodontal disease, and anti-inflammatory cytokines, meaning those are the ones that will kind of stop these inflammatory processes and look into the homeostasis of the tissue. And then here we measure more specifically into IL-2, IL-10 as anti-inflammatory, and IL-17 and interferon gamma. And if you remember, it would attack inflammatory processes. It means that it would lead to destruction of the bone eventually. So what we look again is that the CD5 knockout, actually, if you look all across the different cytokines that we measure, there is an increased number of cytokines produced by CD5 knockout compared to the wild type, meaning there is still a modulation of the CD5 in the production of cytokines. Finally, we wanted to see if the transcription factors associated to those that play an important role in periodontal disease were also kind of modulated over time. And we'll look into this transcription factor. The CSF1 is a colony factor affecting a lot of cells associated to inflammation. This one was upregulated. This one was upregulated as well. And the RAD ligand, interestingly, was just upregulated in those cells that were unstimulated. So what we could see in general was that CD5 can modulate those immune responses. Perhaps more specific data would needed. As I said, the oral microenvironment is not a microenvironment very easy to replicate, but it has the potential to do. CD5 wild type express a higher frequency and activation in T cell differentiation, and CD5 knockout have the tendency to produce higher amounts of cytokines, pro-inflammatory and anti-inflammatory cytokines, and also have the higher upregulation levels of transcription factors associated to periodontitis. I would say that this protein has a lot of potential. This marker has a lot of potential. Future studies should focus more into the regulation that CD5 is able to do on T cells. Now, I really want to thank the Rosemont University College of Dental Medicine and Clinical Outcome Research and Education for the support they've been giving to this study. And also want to thank one of my students that helped me a lot in this project when he was part of the school. He graduated this year, Darlene Cardone, as well as the M Bio department at BYU and all the group and that works in the Weber lab that's been helping tons with this project, especially Carlos Moreno. Thank you so much.